Hello, good morning everyone. Today I'll be presenting our recent work with Dr. Hussein Aziz's team exploring the molecular differences between male and female germline stem cells with a focus on the roles of c-KIT and GFR1 signaling in female germline stem cells, or FGSCs. This study sits at the intersection of the stem cells biology and bioinformatics and aims to contribute to our understanding of sex-specific reproductive mechanisms, particularly with the potential applications for regenerative medicine. For decades, it was believed that females are born with a finite number of ovocytes that gradually decrease over time, and there is not cells known as stem cells in female germline body or organs. But the discovery of female germline stem cells has dramatically shaped this view. Unlike previously thought, female germline stem cells appear capable of generating new ovocytes and challenging the long-standing dogma in reproductive biology. In contrast, the spermatogonal stem cells, known as SCs, males, have been long known to support continuous sperm production throughout adult life. Despite their functional similarities as the germline stem cells, both are germline and both are stem cells, but FGSCs and SSCs may rely on distinct molecular pathways, and understanding these differences is essential for any translational application or any other regenerative medicine application. So the main objective here is to compare female germline stem cells with SSCs and to see how are they different, what pathways are similar, what pathways are different, and how they are doing their cellular, molecular migration, and stem cell maintenance pathways. So here we are zoomed on the c-KIT gene or KIT protein. So a key regulator of germline development is the c-KIT receptor. It is a membrane-bound receptor tyrosine kinase. It is critical for survival, migration, and proliferation. In SSCs, c-KIT typically downregulated in undifferentiated state known as undifferentiated spermatogonal stem cells that will differentiate and become differentiated spermatogonal stem cells. This helps maintain their stemness. But interestingly, as we figured out, in female germline stem cells, c-KIT expression persists, and it has a positive fold change in compared to spermatogonal stem cells, suggesting it might have a distinct extended role, possibly related to differentiation or maintenance. Here, we use the public available data on geodatabase GSE3119 on mouse 422 platform. We use GO2R to conduct the differential expression gene analysis. The outcome of the GO2R was gave to R, and here we can see the Wilkin plot of the analysis showing the most downregulated and upregulated genes along with non-significant and significant but with low fold change genes. So we gave these differentially expressed genes, the list of differentially expressed genes, to a string database. So we can have the protein network of these genes for downstream analysis. But the problem is the output of the string database is a very, very dense network, and it is very hard to have a biological meaning of this. So we gave this network to Cytoscape, and filtered it based on network parameters, degree, centrality, eigenvector, and bitfitness, and filter the proteins with the most number of these parameters. And we have a network of 69 hubgenes that we can use for downstream analysis for biological meanings. So we gave this network to Gephi app to have the distinct modules or the protein, to have a distinct protein modules. Why we did that? So as we know, proteins work together to drive a specific signaling pathway or a specific job in the cells. So this network of 69 genes has distinct protein modules. Every module works together. So if we want to have a biological meaning of the 69 hub genes, first we need to filter them and find the distinct protein cluster or modules. By this, we can split the genes on different parts, and we can have a look deeply in each separate module instead of going and seeing all the module together, all the network together. This enables us to have a very, very more view of the genes and pathways in each module or in the whole network. So after we gave this 69 hub network to Gephi app and conducted the modularity algorithm on that, we got 4 distinct clusters. And c-KIT is in the green clusters with proteins like SRC, IGF2, Wnt5A, and PRFA1. After we have specified this cluster, we need to see what is the meaning of this cluster, what pathways does this cluster do, so we can predict the KIT and its partners in female germline stem cell and what they do in a cell. So we gave just proteins of the green cluster to enricher database and conducted enrichment analysis. On the left table, we see the output of wiki pathway enrichment analysis, which identifies EGFR tyrosine kinase inhibitor resistance as the most significant signaling pathway that these proteins, I mean KIT, SRC, MDK, do in female germline stem cells or in whole in these kind of stem cells. On the other hand, the reactome enrichment pathway output specifies other signaling pathway like PI3K-AKT signaling, GRET signaling, MAPK1, MAPK3 signaling, which needs more research on this field, and we see that what each of these signaling pathways do in a cell. Moving forward, we aim to validate these findings in the female germline stem cells because these are all bioinformatics and these are in silico predictions. So we need to validate these findings in a lab, particularly the c-KIT EGFR interaction. We want to extend our analysis to a single cell RNA-seq analysis and see that what are the c-KIT and other related pathways do in a single cell scale. So in conclusion, our study demonstrates the c-KIT and EGFR1 signaling are central to distinct molecular identifier of female germline stem cells. Of course, there are some other signaling pathways like PI3K-AKT MAPK family signaling cascades, and they need more analysis and more in vitro or in vivo experiments on those. This work enhances our understanding of sex-specific germline regulation and opens exciting avenues for both basic research and therapeutic innovations. At the end, thank you for your attention. I'd like to acknowledge Dr. Hussein Azizi, the public data resource that made this research possible, and of course, the GlobalMeetx team. Thanks.